From part 1, we discovered how to culture bacteria ensuring there is no contamination. In this video, we're going to look at how we can calculate the size of our newly cultured populations. A bacterium has a mean division time of 20 minutes. Starting with one bacterium, how long do you think it would take to produce 1 million bacteria? Pause the video and see if you can work it out. Did you think about 6 hours 40 minutes? The working on the right requires mathematical knowledge of logs, which you don't need to worry about learning. The table on the left works well enough for us. I'm just showing you this for anyone that does know about logs so you can see them being used in real life. What about this question? Pause the video, work it out and click play when you're ready to check. Did you get 8 cells in about 5 hours? Bacteria are becoming resistant to antibiotics, and so choosing the right antibiotic for the specific bacteria is very important. A recent report suggested that by 2050, 10 million people worldwide may die every year from diseases we can no longer cure due to antibiotic resistance. This agar plate with our bacterium on has discs with different antibiotics and concentrations placed onto the bacterium. After time, the colony now looks like this. To measure the area of this clear zone, we need to find the diameter and use the area of a circle formula. So we measure the diameter of the clear zone, which is 2 centimeters. If the diameter is 2 centimeters, then the radius is 1 centimeter because the radius is half of the diameter. So the area of our clear zone is pi times 1 squared, which is 3.14 centimeters squared. Can you work out the cross-sectional area of this colony? Pause the video, work it out and click play when you're ready. Did you get 12.6 centimeters squared? So now we know how to calculate the number of bacteria in a population after a certain time, using the mean division time and listing out the number of bacteria and time in a table. We can also calculate the cross-sectional area of colonies or clear areas around colonies using the area of a circle, pi times r squared.